Our digital media presentation will review new methods and technologies in understanding Huntington's disease.
Neurological disorders are diseases of the central and peripheral nervous system and include up to 600 diseases. Some causes include bacterial, viral, fungal, and parasitic infection, or the immune response to these pathogens. Huntington's disease is a progressive neurological disorder that is said to affect about 5 people in every 100,000 and is characterized by uncontrolled body movements, loss of cognition, and emotional complications. There are 2 forms, being adult onset, which is the most common, and juvenile onset. The condition occurs due to a repeat in the CAG trinucleotide in the Huntington gene, which is responsible for making the Huntington protein. This protein is thought to play a role in neuron formation and normal brain development at the prenatal stage. Early signs include depression, irritability, small involuntary movements, poor coordination, and difficulty learning new information. Some people also experience involuntary jerking movements called chorea, and as the disease progresses, they become more pronounced. Difficulty walking, speaking, and swallowing, as well as a decline in cognition and reasoning, are also symptoms. People live for about 15 to 20 years post-onset, and genetic testing is used to identify the mutation, and this is either diagnostic or predictive. There is currently no way to stop the disease, however symptoms can be slowed to aid the individual in being able to continue normal functioning for as long as possible. Medications such as dopamine blockers can help reduce the abnormal jerking movements and behaviours.
Come in. Hi there, take a seat.
Sorry, this has been happening a lot.
That's alright. Thanks for coming in today, Jerry. As you know, you're here for your results of your genetic testing. Unfortunately, the results have indicated that you do have the genetic markings for early onset of Huntington's. Look, there definitely is hope. From my knowledge, there are 2 promising technologies emerging. The first one is a type of gene therapy. Alternatively, we also have pharmacological intervention. Well, Huntington's is quite a complex disease to treat because of its multitude of symptoms. What we can do in the interim is provide medications that help treat the symptoms depending on which are most prevalent. For now, we can provide dopamine blockers to alleviate the involuntary movements and olanzapine to help keep the mood swings at bay.
Our understanding of Huntington's disease has been greatly advanced through genetic research to achieve genetic therapies as treatment options for patients. Research regarding the mutation in the Huntington gene, HTT, has allowed opportunities to suppress the HTT gene, which could reduce pathogenic protein expression. Development of therapies are focused around 2 areas: the ability to use mRNA or DNA as mechanisms to lower this gene's expression. However, due to the increased number of studies in this area, 2 specific examples will be discussed. uniQure, a genetic therapy company, has decided to develop a vector called AAV5 carried a microRNA, which is a small molecule splicing modulator, which has a specific role to silence the Huntington gene. Its ultimate goal is to inhibit the production of the mutant HTT gene. The research was conducted on animal models which illustrated sustained reductions of the mutant Huntington gene in the deep structures of the brain. However, despite this promising result, the human brain is significantly larger compared to that of other animal models, so its ability to diffuse into the structure of the brain will vastly differ. Secondly, the use of DNA therapies has the potential to have greater effects, which include affecting all components of the HTT gene and could potentially lead to germline treatment which could benefit future offspring of patients. However, there are several ethical considerations which need to be considered before going down this route. Zinc finger proteins are structures which occur naturally and bind specifically to the DNA structures and can be created synthetically using DNA targeting therapy compounds. Research has noted the capability of zinc fingers to be capable of targeting genome editing. Yet, this process ability to be precise and predictable is lacking. However, with the addition of transcription factors, their reliability improves for prospective therapies. However, like any treatment, the biological response needs to be heavily considered. The introduction of zinc fingers has the capacity to trigger an inflammatory response and immune response reactions. Potential pharmacological treatment options include histone deacetylase inhibitors, also known as HDACI, which are chemical compounds that inhibit expression of the enzyme histone deacetylase. This enzyme functions to remove the acetyl group from amino acids that sit on the end of histones during DNA replication, which allow the histones to wrap the DNA tightly and suppress transcription. This process is biologically activated and important for proper functioning. However, in patients that have Huntington's disease, transcription of DNA is greatly disrupted by the mutated version of the Huntington protein.
Gene therapy is an emerging field, which means that further research and clinical trials are required to ensure the safety and effectiveness of these treatments. Other interesting areas of research include drug treatment developments and the impact of early detection and intervention for the prevention of Huntington's disease.